Hello, Socrates at Saddlebrooke. Today I'm broadcasting from the beautiful Mountain View Country Club near corporate headquarters here in Saddlebrooke, Arizona. And I'm pleased that so many have been coming out for these off-hour lecture series, this last one on the origin of life. Last time I shared with you the startling recent discoveries about the specified information that's along the DNA spine here. It's a language and computer processing that takes place from that language to direct what's going on in each and every cell, our 100 trillion cells in our body. Intelligible information. Just a couple of years ago, it was considered junk DNA on here. Only a few of the genes were involved in directing protein building. Now we think it probably is all of them, at least 80% of them. It's intelligible information. It's just a couple of years being considered junk by evolutionary biologists. It's not junk, it's encrypted, it's coded, specified information that directs vital cell functions. Evolution fails to explain, even in principle, just how that level of information could have been pre-programmed into the cell. Now today I'm going to share with you the engineering design of the cell and why that too is also inexplicable by evolutionary processes. Even the evolutionary community agrees that the cell appears to be designed, but then they say it's the appearance of design. It's not real design. Well, is the cell engineered or is it evolutionary? They say it's an illusion of design and not real. Well, is the cell designed or is it just an illusion? How can we possibly tell the difference? Well, I'm going to put forth a simple test that Darwin himself put forth to test theory against research results so we can decide for ourselves. We'll point that way toward real design. We'll point this way to apparent design. And at the end, you be the judge. What direction do you think the scientific evidence points? Now, we pick up my talk with part 4 and the final lecture on the origin of life, and I've entitled it, The Engineering Content of the Cell. Great seeing you. I just love doing this with you all because, like Dick said, the second part is a discussion and question and answer and so forth, and the discussions are the best part of this. But I'll lecture for about 45 minutes and talk about our main topic, which is the information and the engineering that is inside DNA. Last time, if you didn't see it, I really encourage you to go online and look at what is in DNA. It's amazing, the information. And today we're going to talk about what does that information do? It directs the machinery that's inside the cell. And I know people were talking about our 3 big questions there. Where am I going? Where did I come from? Why am I here? And that reminded me of a story with Dr. Albert Einstein when he left Princeton to go on a train ride. Dr. Einstein he bought his ticket at the train station, and it tells you where you go. And has Ardmore, Pennsylvania, whatever, $22. So he's sitting in a seat, and the train conductor is coming through, picking up tickets, and somebody in front of him can't find their ticket. The ticket goes up in a little clip in front of the seat there. And he said, I can't find my ticket. And he's crawling around the floor, and the conductor said, well, you have to buy another one. Comes back to Dr. Einstein. He doesn't have his ticket either in the clip. And he said, I can't find my ticket. And he said, well, that's all right. We know who you are, Dr. Einstein. And he keeps on going back and so forth. And he said, Dr. Einstein's down on his knees looking for I can't find my ticket. He said, it's all right, Dr. Einstein. We know who you are. He said to the conductor, I know who I am too. I just don't know where I'm going. So knowing where you're going, where you came from, and is probably pretty important for all of us. So we're going to be talking about the living cell. The cell is the most fundamental building block of all living things. All living things have cells.
okay? Even plants. Plants and bacteria have cells. We have 100 trillion cells. Pretty incredible little thing. So all living things are made of cells. What do the cells do? Well, they acquire energy from the outside or from nourishment. They use that energy to live. That's how they live. And they cause living things to grow. You grow and develop. The most amazing property is they self-replicate. What does that mean? Can you imagine if you had something that's, especially money, wouldn't that be wonderful to have money that self-replicated? But the living cell does this. So we have to ask ourselves, how did something living come out of something not living? Pretty important question. We looked at what's inside the cell. Last time we saw that DNA is inside the cell. What is DNA? Well, DNA is a biological molecule, and it contains, here's the key, it contains intelligible information that directs the growth of the cell. It directs the growth of the whole organism. And DNA replicates itself, passes that on from generation to generation. That's how we get children. Well, there's one thing we do before that to get children, but that's all right. We're not talking about that. We ask ourselves, where does that information come from? And in our everyday experience, the kind of information we're talking about comes from mind. So we're going to try to connect those two today. Does a cell exhibit engineering? Well, what in the world is engineering? Well, those of you with technical background don't have any trouble with this one, but just think about defining it. It's the art and science of building things, of building structures, building machines. And when it applies to computers and information systems, it's designing and programming information systems. How do you know something's engineered? Reminds me of a Supreme Court case back in the 60s when they were trying to define hardcore pornography. And one of the justices kept on trying to define it. He says, listen, I can't define it anymore, but I know it when I see it. Well, engineering is kind of like that. Is that engineering? Maybe. Could be an artist rendering for all I know. I mean, all we know right now. I mean, first of all, we have to determine, is it real? Does it function? Does it do functional things? Does it have a purpose? Is it designed? If we could answer all those questions, then we could say whether it's engineering or not. Or is it just a random collection of parts? Something just thrown against the wall and looks like engineering, looks like it could be engineering. How about that? Is that engineering? Or is that a piece of art? What do you think? What do you think that is?
Both. According to this class, it's engineering.
That's a cell. Amazing photograph, isn't it? But even looking at that, when you say it's amazing, is it doing anything? Is it operating? Is it being directed? Is it performing function? We don't know that yet. How are we going to do that? We're going to check it out. We're going to say, does it exhibit the qualities of an operational system? Does it exhibit function of being designed for a purpose? Is the information that it contains directed, or is this just an illusion of apparent design? Let's take a deep dive into the cell and find out. Let's first take a walk through the Boeing facility up just south of Seattle. I don't think anybody would object to that being engineering at an extreme level. 100 acres, 50,000 people at the time that was at its peak capacity. 50,000, it even had its own zip code. Look at all those processes that are going on the floor. Many of them mechanical, some of them intelligible by intelligent people, directed by intelligent information. Well, this is engineering at its finest. But how about the cell? Well, I'm going to let Dr. Stephen Meyer give a little insight into what the cell looks like from the inside. We have a book back there called Signature of the Cell. Tom has it. Somebody might want to borrow this particular one. He's the author who will take us inside the living cell.
In 1957, Francis Crick first proposed that chemicals called bases along the spine of the DNA molecule function as alphabetic characters in a written language or digital characters in a machine code. This animation shows how this digital information directs protein synthesis. First, a large protein complex separates the tightly wound strands of the DNA to prepare it to be copied. During this process of transcription, a protein complex called a polymerase produces a single-stranded copy of the original instructions. Here we see this copy, a messenger RNA molecule, being constructed inside the polymerase as individual bases are positioned and added to the growing strand. Now we see the polymerase in action from the outside as it spits out the messenger RNA transcript. Next, this RNA transcript approaches and passes through a molecular machine called the nuclear pore complex, an information recognition device that controls the flow of information in and out of the cell's nucleus. Now we see the genetic assembly instructions on the messenger RNA approaching and arriving at a two-part chemical factory called a ribosome, the site of protein synthesis. As the messenger RNA transcript passes through the ribosome, the process of translation begins. During translation, a mechanical assembly line builds a specifically sequenced chain of amino acids in accord with the instructions on the transcript. These amino acids are transported from other parts of the cell by molecules called transfer RNAs, which link specific sequences of bases to corresponding amino acids. The sequential arrangement of the amino acids determines the type of protein constructed. When the construction of the chain is complete, it is transported to a barrel-shaped machine that helps fold it into the precise shape required to perform its function. After the chain is folded into a protein, it is released into the outer cytoplasm to do its job in the cell.
Does that remind you of the Boeing facility there? It has the same things that we see out on the floor there, the Boeing facility. We see power plants, we see manufacturing facilities, we see transportation systems, we see waste treatment systems, staging. All these have biological names, but basically they're doing these engineering functions. This system fits together just like the Boeing manufacturing floor. This is engineering and information at the most elegant level. These little devices inside the cell there called organelles, just like we have organs, the cells have organelles. They're biochemical machines made of biomolecules. They're engineered to work as a system on a nanotechnology level. Each organelle has a specific purpose and a specific design, and it does a specific function. And it's directed by the information that's in the DNA molecule that we discussed last time. So what we're seeing here is something far more complex than the manufacturing process of the Boeing 747. In fact, the instruction manual that we looked at last time, the 3 billion characters that are inside the cell, we showed that would be a 350-foot high instruction manual if I printed it out on my computer. The instruction manual for Air Force One 747 cost $84 million, is 100,000 pages, and we're talking about just one cell. So inside the cell now we're finding power plants, and look at the scale there, the city blocks. Something as sophisticated as that is contained in the mitochondria. We find manufacturing facilities, the ribosome that he talked about. One protein makes a protein, makes the right proteins. There's hundreds of proteins, manufacturing factories in there. Transportation systems, it makes its own highways. Microtubule highways that transport the devices or the manufacturing from one place to another, has its waste treatment facility. It's a complex of anything on an engineering scale that we would put together. Staging and storage areas, chemical treatment areas, shipping stations. I mean, you get the idea. And it's all controlled in the control room by this DNA. Totally integrated. Precisely controlled. So what's a rational conclusion here you could make? One rational conclusion is the cell is an engineering marvel. This manufacturing process has been engineered. The systems are intentional. Each has a purpose and a design. The systems are integrated together to produce life. Produce such a system in any kind of human way would require engineering, require intelligence. So what does modern evolutionary theory tell us about it? Well, it says, yes, it does appear to be designed, but it's an illusion. Nature assembled this cell by herself, accumulating accidental, small, undirected steps over a long period of time. The cell manufacturing process is unplanned and undirected, according to modern evolutionary theory. The modern molecular machines that we see are created by evolutionary process called random variation working through natural selection. So no intelligence is required. Our top biologists declare that the design that we see is an illusion. Let me quote. Richard Dawkins, one of our prominent evolutionary biologists, said the living results of natural selection overwhelmingly impress us with the appearance of design, impresses us with the illusion of design and planning. Francis Crick, one of our most illustrious Nobel Prize-winning scientists who discovered the structure of the DNA molecule, says, biologists must constantly keep in mind that what they see was not designed, but rather it evolved. So the question is, design is an illusion. Well, let's test that claim. I mean, Darwin himself established the test for that claim. He said, if it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, then my theory would absolutely break down. So the big question is the design real or is it an illusion? Whatever it is, it has to be at least as powerful as the apparent engineering that we think we just saw. The core of evolutionary theory is this concept of random variation and natural selection. So let's just take an overview of that. Random mutations occur in nature. We know that to be true. We're just going through one right now. Mutations take place. And they can be detrimental, they can be neutral, or they can be positive. We do have positive ones. We have positive mutations when we have adaptation. We know that sheep that are grown in northern Scotland grow longer hair than ones that are grown, raised in the Sahara Desert, have shorter hair. We adapt to our environment. So we know that some beneficial mutations occur. Antibiotics in the lab. We know we can make antibiotics by mutating and even randomly mutating certain cultures. But beneficial mutations are passed on by reproduction. This is the concept from one generation to the next generation. And when they're passed on, they can provide a survival advantage. Make sense? Natural selection is a process has no direction, no design. It's a random process. But generation after generation, taking on a cumulative random mutation can build up a population's fitness in nature. And that's why it's called natural selection. It's not artificial selection like when we breed dogs or breed pigeons or something like that. Nature can do some of this. Called survival. That's why it's called survival of the fittest. But when we get down to the cell level, that's at the gross level. We get down to the cell level, how does evolutionary theory explain the cell? Well, we've been through this in a couple lectures before. On early Earth, unknown chemical processes for dead chemicals turned dead chemicals into life's first building block. But those processes are unknown. Molecules must self-replicate, make themselves. But we have a known process that does that, according to evolutionary theory. There are no known natural selection mechanisms to create the first cell. Plenty of talk, plenty of theory, plenty of experiments, but none of those theories hold up. All theories have failed. And the biggie is that there are no detailed biochemical accounts for how natural selection occurs at the molecular level, even in principle. Well, I was hoping for more information than that. But we can't be too hard on Charles Darwin. I mean, after all, in 1859, when he came up with his theory, they didn't know what a cell was. They thought it was just a blob of jello-like substance that was easily explained, and it could grow and create life on its own. But a lot has happened in 168 years. Last 50 years in particular, we have technology, like we have the Hubble telescope that has taken us to the edge of the universe and has given us some thoughts. Now we have Hubble telescope microscopes that allow us to dig deep and look into what the cell is and see that incredible picture. We have other technologies, nuclear magnetic resonance and X-ray crystallography. And I was reading just recently, 2021, we came up with a technology called RD Sprite, which allows us to look inside the nucleus, the little tiny nucleus where the DNA is that directs the protein synthesis. And we can see what's going on inside there. It's pretty incredible. So that's the picture of what's happened between 1859 and 2022.
Yes.
The result is evolution is a theory in crisis. A theory in crisis. It has not been shown by observation and experiment how evolution works at the molecular level. In fact, there's no publications in the prestigious scientific literature, journals, or even books that demonstrate how it works at the molecular level. Many claims at a high level, many claims that such evolution must have occurred, but none are supported by pertinent experiments or calculations at the molecular level. There hasn't even been a meeting or a paper that describes in detail the evolution of complex biochemical systems. So, increasingly, many skeptics have reached and researched this matter for themselves. In fact, over 1,000 doctoral scientists now have gone public to voice their skepticism. They've signed on to a public statement, which is putting themselves at risk because this is not the mainstream scientific conclusion. The mainstream scientific conclusion is still Darwinian evolution. But they came out and said, we are skeptical of claims for the ability of random mutation and natural selection to account for the complexity of life. Careful examination of the evidence for Darwinian theory should be encouraged. Well, I certainly agree with that. Right now, there's a lot of trying to understand within the biological community how this could happen on a natural basis. We're going to hear from two of these scientists that were evolutionists and have come over to understand that we're skeptical of this. And they've performed experimentation to demonstrate why they're skeptical. First person we're going to hear from is Dr. Michael Behe, a professor of biochemistry at Lehigh University. He shook up the community in 1996 with his book, Darwin's Black Box, The Biochemical Challenge to Evolution. Let's hear from his story. Observed at the molecular level, these machines. One of the machines that really struck him was when he looked into a biochemical textbook and it showed the tail that's on the end of a bacteria. the tail wiggles, and that's how the bacteria goes around and collects its food for its nourishment. And he's looking at this and he said, that's no random collection of parts. That's an outboard motor. That's designed. When you look at that, I mean, there's no chance assembly of parts here. And that really convinced him that he should be looking into another way of understanding how that got to be. So he was very upset about his educational program because he never heard of these things. This never was brought to his attention before. I want to show you what this bacterium flagellum looks like.
The flagellar motor is the one that Behe made most famous. It's a rotary engine that powers a whip-like tail, a protein tail, that functions like a propeller, and it moves the bacterium through liquid, enabling the bacterium to essentially track down its food supply. And this little machine includes a rotor, a stator, a drive shaft, a U-joint, bushings, bearings, and a whip-like tail that functions like a propeller. And the machine in some bacterial systems turns at 100,000 RPMs in one direction and can reverse direction on a quarter of a turn and turn 100,000 RPM in the other direction.
Well, I think you get the idea. That's not a random collection of parts. And it requires some 40 proteins. Protein is a very complex chemical. We talked about how that was formed last week from the DNA information. It has 40 different proteins that have to come together at one time for the machine to function. It's an outboard motor. Now, motor could not possibly have been formed by numerous successive slight modifications. That's one of the criteria that Darwin said. The motor can't wait for evolution to supply the next part. Behe coined the term irreducible complexity. So let's look at Darwin's criteria for what demonstrates whether Darwinian evolution is true or not true. He's talking about a complex organ, or an organelle in our case. We're talking about the bacterium flagellum. That's just one machine in the system. And he said if it could be formed by numerous successful slight modifications, well, all the necessary parts and specifications have to come together at one time. You can't have half of the parts waiting for another part, especially when we're talking about this being produced over a long period of time. And he said his theory would absolutely break down, and Behe has, in his view, demonstrated that flagellum is irreducibly complex, so the Darwin theory has to break down. But let's break it down even simpler. Let's take a household machine that we all know, a mousetrap, okay? Only has 6 parts. There's a spring and a holding bar and a catch and a platform and so forth. It needs all those well-matched parts at one time to work. And they have to be precisely engineered. I mean, if that little catch is off by a couple millimeters, you can't spring it back and hold it. It has to be precise. If any part is missing or off spec at all, the machine stops functioning. There is the flagellum with 40 complex protein parts. Any spec that's not met or not available at one time, it won't work. So the cell, in comparison, is astronomically irreducibly complex. Natural selection cannot deliver. Evolution is a step-by-step, trial and error process over long periods of time. That's the definition of it. It does not deliver all the matching parts when they're needed. In addition, there's no purpose or direction in natural selection. So if it was 40 proteins, it doesn't know it even needs 40 proteins. There's no blueprint for it. It's a blind, random process. Now, and if some parts are missing or they're not working, it doesn't result in less function. If you have a mousetrap with 3 of the 6 parts missing, you don't catch half the mice. You need everything at one time. If a living system doesn't have all its well-matched parts at one time, what happens? It's dead. It dies. For natural selection to work, it must even have a previous living organism that reproduced to pass on the selective advantage, as evolutionary theory says. It has a selective advantage. There is no evolutionary parent. There is no parent. Where'd the parent come from? So anyway, trial and error, Darwinian evolution cannot overcome the requirement of irreducible complexity. So how has the evolutionary community responded to this? Well, it's been quite interesting to watch this over the last 20 years. Since 1996, they've been trying to answer Behe and others who have done experimentation in this. And I'm talking about scientific organizations that are committed to evolutionary biology now. Their journals have not been able to successfully answer him. There's plenty of theoretical discussion and plenty of firestorm of criticism. We know from all the scientific debates that go on with other things like climate change and whatever, there's two sides. You have to hear two sides to get anywhere. You can't just listen to one side. And if you go out to the internet, you'll see substantial criticism of Behe's work. But has it been read? Has it been addressed? He's addressed all these criticisms, by the way, and the same criticisms keep on coming back. So be careful what you read. Read both sides. Read the information on both sides, like we have to do in our political process. It can't just be, yes, I'm right and you're wrong. The internet, popular press, and media respond with a lot of high-level hand-waving. Unfortunately, a lot of bullying that goes on. A lot of bluster, making assertions that go way beyond the evidence. And unfortunately, personal attack. Behe responded to this criticism in a book. I'm not sure, Tom, if I have this one back there. Ah, you do. Okay, that's a good one. That's a new one, too. He took all his answers, all the criticism that he's been getting from Nature, scientific journals, the high-level organizations, plus even the blogs of evolutionary biologists around the country, and has answered them. And then he compiled it because they say, Michael, you do not respond to our criticism. He says, yes, I did, in such and such a point. Anyway, there's a book there called A Mousetrap for Darwin, where he takes the criticism in the paper that has been levied against his position, and he answers it. So I say, read it. Evaluate the arguments for yourself. When I get into discussion with people, what I usually find out, there's mischaracterizations and straw men in this kind of discussion all the time. People hear, they think they know what irreducible complexity, for example, is, or intelligent design, and they've read something that is from their side of it, and they think that's really what the conclusion is. We all know, even from our own biases, that we look for information that supports our position. So the best thing to do is read the source material. And it's no longer just in high-level scientific journals. It can be explained and understood at the mousetrap level. So I encourage you to do that. And in discussion here after the talk, I'd like to keep that as a goal too. I mean, let's just discuss what's going on from both sides. So, in a review, what's the engineering content of the cell? The cell is an engineered system of complex molecular machines that exhibit design and purpose. Here's a biggie. Design and purpose can be scientifically detected. Have you ever thought about that? How do you know something's designed? What's the scientific approach to it? Got to come back next month when we start discussion on that. How do we scientifically understand if something's been designed or not? The cell is irreducibly complex. Is it a parent? An illusion, or is it a fact? Darwinian evolution, chance plus time plus natural selection, cannot engineer molecular machines. Complex integrated machines are created by intelligent agents. So what's the status of mainstream evolutionary biology? This is from a textbook, my textbook, Evolution, Douglas Tuma, 2009. Let's read this carefully. Natural selection is the most important, and this is what we teach all our students. This is exactly what we teach our students. Natural selection is the most important concept in the theory of evolutionary processes. I agree, that's their position. It is surely the explanation for most of the characteristics of organisms that we find most interesting, ranging from the origin of DNA to the complexity of the human brain. Now that's quite a statement. That goes beyond even what I showed here. I was only talking about proteins. I was talking about the complexity of DNA. And I certainly wasn't talking about the complexity of the human brain. I mean, we are going to do that, by the way. We are going to go and see the neurological processes that are going on here. So my question to you all, and for discussion with your friends and so forth, is does the evidence, the hard evidence, support that claim? As you can imagine, we're talking about and with very, very smart people here that are looking at this. I'm just a researcher. I'm like a journalist. The research is what the literature is, and hopefully I'm looking at it from both sides. And I certainly have my own opinions, and you know what they are. In the community, there's a growing dissatisfaction within the mainstream. And here's just the last few years. A group of 16 evolutionary biologists got together and said, we got to do something. This is not holding up. Darwinism is not holding up. And they're called the Altenberg 16. 2011, there's a group that is looking to nature and how nature is engineered. They agree nature is engineered. Don't know what the mechanism is, but they believe that there's a natural mechanism in nature that engineers these cells. Now it's moving into the Royal Society of London. That's big time. That's like our National Academy of Sciences in England. They held a conference to try to see what are we going to do about this, because Darwinian evolution is failing as a theory. We need an alternative conceptual framework. 2016, a subgroup of that group met and said, they were even more pointed. They said, current evolutionary theory fails to explain the origin of new anatomical structures. That means the origin of the species. New anatomical structures. So a paradigm shift is definitely taking place. And as we know, paradigm shifts occur in science all the time. We started off with a paradigm shift when we first talked about the beginning of the universe. Beginning of the universe 100 years ago, it was static, it was infinite, and it was just always there. Now, what's the theory? Big Bang Theory, right? It started a finite time ago, and it's accelerating. It's not even an explosion because it's not decelerating, it's accelerating. That's what 100 years from now, what's the theory going to be? Mm-hmm. So this is time for revision in evolutionary thinking, in my view anyway. So what I'm going to do in the next session, and here's where we're really going to have some fun, is we started off this whole series saying that science and faith are two things that need to work or at least inform one another. And the reason is both are seeking truth. You can either seek truth from a religious position, you can seek truth from a scientific position. Both are seeking the same thing. Why are they so divergent? Why don't they try to inform one another? They should come out eventually when we know more, when we really know the right answer, when we're down to the right understanding of things, they should say the same thing, right? What we physically observe and test and so forth should be the same thing as we believe by our faith. So if you're a faith person, you should know why you believe what you believe and be able to defend that. If you're totally a science person, you should understand the philosophy of naturalism. Naturalism, which your theory depends on. We're going to discuss this. So we're going to get into the evolution creation debate, maybe from a different angle than you ever saw it before. I just want to inform everybody of what the main premises are from evolution, creation, intelligent design, which is a new science now, which Behe and Stephen Meyer represent. And then there's a whole group called theistic evolution. They try to combine the two. They say, yep, I want my cake and eat it too. I want to have believe in Darwin, and I want to believe in biblical creation, and they put those two together. Those are pretty much the broad categories of the groups of the views. But what I want to extract from each of those views is what is being said that's scientific, observable, testable, falsifiable, so forth. What is being said that's from faith? Darwin had a lot of faith. He went about his theory of evolution from a faith position. We'll talk about that. It's not generally discussed. What is the philosophy that you start with? What's the worldview you start with? I mean, if you start as a biblical creationist, you're going to end, and that's where how you interpret information. If you start as an atheistic evolutionist, you're going to interpret the evidence from that. But there has to be some core of information that can be discerned as, yes, that's scientific, that's worldview, that's the faith. So make sure you come back for next month when we start that discussion. So with that, I'm going to conclude session 4, which is the origin of life. We talked about the origin of life, the very first life on Earth, and where that came from. How did life originate from dead chemicals? What are the theories about that? We talked about DNA and the information. Now we talked about the engineering that's in it. And the good part about this, I'm putting these up on videocast. So this will be up in a few weeks, and it'll be without all the interruptions. And it's available as a 5-part series on the origin of life. So please go to the club, www.Socrates@Saddlebrooke.club.
When I finished the talk, there was strong reactions from the audience in both directions. Now, those who were educated strictly in the biological sciences were shaking their head, no way, Jose. That doesn't agree at all with what's being taught, even through our doctoral programs. And it isn't even considered science by the Academy. I mean, after all, Darwinian evolution has been the mainstay theory in biology for over 150 years. And it's famously known and said that nothing in biology makes sense except in the light of evolution. Evolution is the overall reigning paradigm of biological origins. So how could some engineering perspective come in now and possibly reveal anything so radically different? But those in the room who were schooled at some level of engineering or information sciences or computers were shaking their head in agreement. They grasped the necessity of getting down to the nuts and bolts and the bits and bytes of how the cell works in order to see how can an evolutionary mechanism actually work at that level. They could see how evolution's mechanism of random variation and natural selection, which we'll talk about in the next series, might not work at the molecular level, even with billions of years, even with trillions of years. Some others, on the other hand, were saying, well, we need to think more about this. Some were having an aha moment. Others, unfortunately, became angry. Behe's insight into the bacterial flagellum was particularly disturbing to them. Comparing a biological flagellum to an outboard motor, that's absurd. But his engineering reasoning of irreducible complexity is not absurd, and it caused quite a stir. Some even went out to buy his book, or were reading his book, Darwin's Black Box, his insight into this bacteria flagellum and comparing it to an outboard motor, and the subsequent answers that he gave to his critics over 25 years in his latest book. Some were obviously shaken, and not just talking about it over a beer at Callahan's, but even openly discussing it in the lab this week. So when that buzz gets back to management, I'm not sure what the repercussions are going to be on future series, but we'll worry about that then. Especially when I start talking about Darwin's theory of the origin of the species. Now, if you wanted to stay afterwards to discuss the entire series we're just finishing up on the origin of life, so I prepared a couple of PowerPoint slides to summarize my conclusion. How did life begin on planet Earth? What is the information content of DNA? What is the engineering content of the cell? Now, we didn't film that portion, but I posted those PowerPoint slides without commentary on the website too. So you can see them by going to the website, clicking on Videocast, number 4, Origin of Life, click on Conclusions. But to get the full treatment, go back and view the whole series, number 4, on the website. In this next series now coming up, and I'm quite excited about that, I'm going to be discussing Darwin's theory of the origin of the species. And I begin in part 1 by discussing the 4 major views regarding how the diversity of life came about on planet Earth. Was it Darwinian evolution? Is it biblical creation? Is it intelligent design? Is it theistic evolution? In part 1, I will review the underpinnings of each of these positions, and which is commonly referred to as one's worldview. Worldview is the glasses that we put on when we look at things, when we see the things around us and how we investigate. Very important to determining how we see evidence, how we interpret evidence, what path of inquiry we follow. I remember one of my professors, one of his favorite things is that, remember, all the world is jaundiced to the yellow eye. So, next time, Origin of the Species, part 1. Keep asking the why questions as well as the what questions, and follow the evidence wherever it may lead. I'm Stuart Orr.